Hi, we're going to use this video to take a look at the structure and function of the organelles that are found in the cell, specifically the animal cell, which is what you find in you as a human. As you know, the cell has a variety of organelles that play essential roles in life, in keeping the cell alive and ultimately in keeping you alive as an organism. And those organelles fit into two categories, membranous and non-membranous organelles. The membranous organelles are isolated compartments that are surrounded by a phospholipid bilayer or membrane. And those membranous organelles include those listed here: the nucleus, the mitochondria, the endoplasmic reticulum, the Golgi apparatus, lysosomes, and peroxisomes. The non-membranous organelles include the cytoskeleton, microvilli, centrioles, cilia, flagella, ribosomes, and proteasomes. Again, these are organelles that do not have phospholipid bilayer membranes surrounding them. If we look at the functions of the non-membranous organelles, the centrioles, or centrosomes as sometimes they are referred to, are the structures that direct the chromosomes in mitosis. The spindle fibers will develop from the centrioles as they move to the poles of the cell to help separate the chromosomes and allow us to make two new cells from one initial cell. Microvilli, cilia, and flagella are all found on the outside surface of the cell. Microvilli are tiny little surface projections, and their role is to increase the external area or the surface area of the cell. That's going to allow the cell to have more membrane in contact with the environment for communication and transport across the cell membrane. Cilia are small hair-like structures that are on the outside of some cells, and their job is to move fluids across the cell surface, and sometimes that also means they help the cell move through fluid. The flagella is more of a tail-like structure that allows cells to move through fluid. Think sperm cells, and the whip-like tails that they have, those are flagella. The ribosomes are two small subunits that come together to make new proteins, and those are mostly made of a ribosomal RNA and protein, and again, they don't have a membrane. Finally, proteasomes, which may be new to some of us. Proteasomes have a role of digesting damaged proteins that are no longer functioning. That breaks them up and allows the amino acids that made those damaged proteins to potentially be used in making new proteins. The cytoskeleton itself is important in helping to maintain the shape and structure of the cell, and also to help hold all the organelles in place within the cell. There are three major components, all of which are sort of fibrous, that are going to connect the various parts of the cell and connect to the cell membrane on the inside of the cell. The microfilaments are mostly actin. There are intermediate filaments, which are a little bit thicker, and those vary by type. And finally, there are microtubules made of tubulin, which also help direct the movement of chromosomes during mitosis and meiosis. So this picture shows some of the components we've just been discussing. You can see these projections on the outside of the cell, which you can also see in this whole picture, are the microvilli. Those are, again, to increase the surface area that the cell has to allow more communication and transport. We also can see the kind of tan-colored microfilaments within the cell. Again, those are also going to help maintain the structure of those microvilli in addition to the cell structure itself. You can also see the intermediate filaments, which are sort of purple in color, and the microtubules, which are tube-like structures that in this picture are denoted as yellow. You can see some other organelles within that are held in place by those filaments. The membranous organelles are the ones that you're probably already more familiar with. The first of those is the endoplasmic reticulum. The ER is, as you can see here, a network of intracellular membranes that help the cell make molecules or do molecular synthesis. That endoplasmic reticulum network does fill up a large portion of the cell, and it includes two different types of endoplasmic reticulum, the rough and the smooth. The structural difference between rough and smooth endoplasmic reticulum is, again, that rough ER contains ribosomes studded in its membrane, and that helps it to do the job of supporting protein synthesis and helping with the packaging and moving of those proteins. And the smooth ER, which does not have ribosomes studded in it, and its job is to also do some protein synthesis, but also it makes carbohydrates as well. The majority of the endoplasmic reticulum in the cell is rough endoplasmic reticulum. And in this picture you can see again these membranes are surrounding these sort of tube-like highways throughout the cell. The next of the membrane-bound organelles is the Golgi apparatus. The Golgi apparatus, also sometimes called the Golgi body, is highly connected with the endoplasmic reticulum. It's going to receive the proteins that are made on the rough endoplasmic reticulum. It's also going to add carbohydrates and lipids, as well as package proteins into vesicles. Some of those vesicles or pockets are going to be secretory vesicles to allow the contents to be secreted or removed from the cell. Some of those vesicles are going to be membrane renewal vesicles, which will help to build the cell membrane as some of it gets wrecked or used. And some of those vesicles become lysosomes, which are going to help to clean out the cell as they're full of digestive enzymes. So as the products are made in the endoplasmic reticulum, which you can see here, they are going to leave the endoplasmic reticulum sometimes in small vesicles. Those vesicles are going to travel to the Golgi apparatus, as you can see in this picture. The Golgi apparatus structure is sort of similar to the endoplasmic reticulum, except it's more stacks than it is networks. The Golgi apparatus, again, is going to produce a variety of things. As you can see in the top here, we have a pocket full of digestive enzymes, or a lysosome. We also have situations where we have membrane renewal vesicles that are going to bind to the surface of the cell membrane, open up, and then become part of that cell membrane itself. And finally, we have our secretory vesicles, which you can see are secreting the red molecules, in this case, to the outside of the cell by being transported to that membrane and then opening up. If you look at that secretory vesicle action with an electron microscope, this is what you're going to see. So again, you can see that vesicle here, which is bonded to the cell membrane, and the contents of that vesicle are being removed. And again, that's exocytosis. The lysosomes, which again are created by the Golgi apparatus, are packets of digestive enzymes, and their job is to fight and kill bacteria. They also clean up debris within the cell, and they act as suicide packets. So when the cell is needing to undergo apoptosis, the lysosomes are going to initiate that process. The next membranous organelle is the mitochondria. You already recognize that the mitochondria is the powerhouse of the cell. That's because it creates 95% of the cell's ATP supply, and it does so by using a variety of proton pumps and electron transport system complexes. The mitochondria has a double membrane structure, so it has an outer membrane that is pretty highly permeable, and then there's an inner folded membrane of cristae, and that's a little bit less permeable. The cristae are surrounded by the aqueous matrix, or the watery matrix. It's filling up the mitochondria, and they're started with electron transport system complexes so that in cellular respiration, the electrons have a chain to go down to ultimately power the making of ATP. Here's a slightly better picture of the mitochondria than the one that I drew. You can see that the inner membrane, or the cristae, are studded with enzymes to make those chemical reactions happen quickly, and the inputs to the mitochondria are organic molecules such as sugar and protein and lipid, as well as oxygen, and the outputs are CO2 and ATP. The final, and sometimes arguably the most important of the membrane-bound organelles, is the nucleus. The nucleus is the largest of the organelles in terms of size, and its job is to be the control center to control cellular operations. The nucleus is what determines the structure of the cell and directs all the things that the cell does. The nuclear envelope, or nuclear membrane, is what separates the nucleus from the cytoplasm, and the nuclear pores are the tiny holes within that nuclear envelope. As we discussed already in class, we do need to be able to transport things in and out of the nucleus, and the nuclear pores allow that to happen. The nucleus is the storage location for chromosomes, and as you can see in this picture, we do have the nucleolus, which is our dense clump of DNA. We also have chromatin, and the chromatin is the decondensed DNA when it's not actually in the chromosome structure. We only see the chromosome structure when the cell is undergoing mitosis or meiosis. Again, we can see the pores, or the holes, in the surface of the nuclear envelope here as well. And this is a picture of an electron microscope image of a nucleus. We can also see the endoplasmic reticulum surrounding the nucleus as well. And the final piece to take a look at is the structure of the chromosomes again. The chromosomes are the nuclear DNA, and the chromosomes are, again, the instruction code to make the organism. And they provide the instructions to make the proteins, which ultimately determine your traits. Each of you has 23 pairs of chromosomes, 23 from your mom and 23 from your dad. Each pair of chromosomes contains similar information, but obviously it's a little bit unique because your mom and dad may not have given you the same hair color gene, for example, or the same eye color gene. In order to create the structure of the chromosome, the DNA is wrapped around histones, which are proteins that kind of hold the structure of the chromosome together. And the decondensed DNA is called chromatin. And again, this is that structure. We can see the DNA itself. That DNA is wrapped tightly around histones, and that is further coiled and supercoiled into the actual chromosome structure that we recognize. In a non-dividing cell, that chromatin is decondensed, so it's not going to be in this nice rod shape. It's going to be unwound, as you can see in this picture. And there you have it, the structure and function of all of the cell's organelles. Hopefully this was helpful. Feel free to email or stop in with questions that you may have.